Johann Friedrich Miescher was able to isolate a substance from white blood cells in 1869, which he called nuclein. Miescher's study, which led to this discovery, involved analyzing white blood cells obtained from pus on patient bandages. In the early 1900s, Phoebus Levene discovered the building blocks of the substance earlier identified by Miescher. He called these building blocks nucleotides and demonstrated that each consisted of a phosphate molecule, a sugar molecule, and a nitrogenous base. The polynucleotide model was developed by Levene to describe the way in which nucleotides were strung together.
In 1951, Rosalind Franklin met up with Morris Wilkins, who was already in the works of studying DNA through X-ray crystallography. Together, their work led to the discovery of the double helical structure of DNA. In 1971, Chargaff discovered that the nucleotide composition of DNA varies among species, contrary to what Levene had hypothesized. He also created what is known as Chargaff's rule, which states that the total amount of purines and the total amount of pyrimidines are usually equal. In 1953, Watson and Crick used cardboard cutouts to discover the three-dimensional structure of DNA. The discovery of DNA has impacted the field of medicine immensely. One thing that the discovery of DNA has led to is genetic screening. The purpose of genetic screening is to see what conditions you may be predisposed to, as well as what conditions you or your partner could potentially pass on to future offspring. This form of screening can be very useful, especially if the genetic conditions you are predisposed to are life-threatening or life-altering.
Huntington's disease is an example of a genetic condition that can be traced through genetic screening. Huntington's is an autosomal dominant disease where the probability of offspring inheriting the condition is 50% with at least one affected parent. Gender is not a factor in determining who will inherit this condition, and as you can see in the example, Huntington's disease does not skip generations. Medical research like the discovery of DNA continues to impact the field of medicine today and has made it possible to screen for a variety of genetic conditions.
Gene therapy is the addition of new genes to a patient's cells to replace missing or malfunctioning genes. The discovery of DNA has led to a possible cure of hepatitis B in patients using gene therapy. Nucleic acid-based therapy can potentially eradicate hepatitis B virus in patients, can bind to and disable the virus. Gene therapy is also used for indicating mitochondrial DNA damage in patients with Huntington's disease.